important time right now when computer scientists and neuroscientists and engineers in general really need to be talking together on many different levels. And I'm going to tell you a story of an adventure that I've had over the last 2 years. And in it I'm going to touch on many of the themes that I've already heard about this morning, giving a personal view, but also trying to look into the future. So if you look at the brain from the spatial perspective and levels of investigation from the molecular up to the entire central nervous system, there are 11 orders of spatial scale here. So part of the problem is not just that the data are heterogeneous, it's that they're taken from techniques that are so far apart from each other that it's hard even to know how to stitch them together. How do you bridge across these levels? And one of the tools that we have, it turns out to be building models at each level and using bridges between the models rather than the data. So that's one of the tools we have, is to actually use techniques from physics and from engineering that have been very successful in the physical world and apply them to the biological world, but it's going to be different. It's not going to be the same, and I'm going to be getting back to that. I just want to illustrate that here, just one example here. When you're doing, you've heard the word connectomics. Well, it turns out connectomics can be done at each one of these levels, looking at the connectivity pattern between neurons in the brain. If you do it down here at the level of individual synapses, and here is an example of a synapse, then it's called microconnectomics, and that's what people like Jeff Lichtman and Sebastian Seung and Winfried Denk are trying to do. They're trying to do at that level larger and larger chunks of tissue. Well, there's another way you can think about connectomics. We know that in the human brain, for example, there are very long-range fiber tracts that span many, many centimeters. And it is possible with new techniques like diffusion tensor imaging to be able to trace them and give a sense for what the long-range communication is like. Well, that's on a very much higher level here, up in the centimeter level, right? And there are going to be connectomic maps that are being developed at all levels using tracers, for example. I'm going to give you an example of that later on. But it's just that we have a real challenge here. It's a multi-scale challenge. So on April 2nd, our president made an announcement which really had, I think, implications for many research groups and for research programs and for a lot of, and that's why we're here. I think we wouldn't be here if it hadn't been for the fact that he chose us. He chose the brain as the grand challenge project that will be his legacy, right? He could have chosen others, right? There are a lot of other grand challenges out there, whether you're from NASA, you want to go to Mars, or whether you're from DOE and you want to build a better battery, right? There's a lot of things that could have had practical applications, but I think that this one captured the imagination because it's a problem, it's something that we all share. We all have, we're all getting older. And memory, and for example, we don't understand fundamental things, like how does it that we learn and remember and forget? These are things that are still being studied at each of the levels. But the key here is an acronym, is the last two letters, I and N, because rather than focus on a disease, a specific disease, and there are so many very, very devastating diseases like Alzheimer's, depression, rather than do that, what the Brain Initiative is trying to do is to develop innovative neurotechnologies, better tools. And the goal is to accelerate research, to be able to compress 100 years of research into the next 10. And that means bringing people who can build tools to work together closely with people who have the problems and the questions that need to be answered. And we're just getting off the ground. But nonetheless, I think that it's heading in the right direction. So I was chosen to be one of the 15 on the advisory committee to the NIH director working group to put together a report. And Francis Collins, the director of NIH, met with us and said, what we want you to do is to think blue sky. We want you to come up with goals. We want you to come up with milestones. We don't expect you to tell us how to get there. But if you tell us what's important and prioritize them, then we'll bring out the best talent and try to get the teams working together. And that was itself really an exercise that lasted for 15 months. We had 6 meetings that were like this one, lasted 3 days, and then we had another 6 meetings that were conference calls. So imagine, okay, this group coming back every 3 months for a year and a half, right? Well, after a year and a half, you get to know each other pretty well. In fact, I learned more about neuroscience in the last 15 months about areas outside my own field than I would have ever imagined because we went into depth by bringing in outside consultants, the best people, 48 outside consultants, patient advocacy groups. We really wanted to get a complete poll of all of the stakeholders. And I learned a lot, not just about neuroscience, I learned a lot about my colleagues, about what they thought about what I did, and they learned what I thought about what they did. Something that, you know, if you stay in your little silo, you don't necessarily, get that feedback every day. But it was extraordinarily constructive in the sense that we did, at the end of the day, on June 5th, come up with a document, 150 pages long, which represents the best thinking that we were able to do. And I have to say that it was really, although it was a team effort, Bill Newsom and Corey Bargman were the ones who really brought it all together. They were the ones who had to homogenize all of our prose and cut, because of course each one of us wanted to increase the part that we thought was the most important, which is always the problem. How many people here have read our report? This is pretty good. Okay, I see that there's at least a dozen people who have read it. So, you know, the purpose really was to provide a way of thinking about the brain from a technological perspective in terms of, you know, where could we make the most progress. But it turns out that the place that really, for whom this has really been a big hit, has been NIH. So program officers, and we have a representative from NIH, they argue amongst themselves about what's important. And you know, they have to decide where to put their programs and who gets the resources. And you know, and NSF program officers probably have similar debates amongst themselves. Well, this is their Bible. So when they have disagreements about, you know, whether this approach or this technique is more important than that, they go and they look at the chapter and verse to see what, you know, what is it we were thinking. The problem is that the field keeps moving, and so this is by, you know, 2025, I'm sure this is going to be completely obsolete, but that's actually our goal, is to try to make progress in ways that may not even have been anticipated. Okay, so here's a Cook's tour of what's in the report, you know, for those of you who didn't read it. And broadly speaking, the idea is to put together big databases. First of all, we don't know how many different types of cells there are in the brain. The place where actually a lot of work's been done in the retina, we think there's on the order of 100, but that is just an approximation because there are many species with many different retinas. But in the cortex, the estimates range from 100 to 1,000, and that's because two cells that look identical, two pyramidal cells, may have very different physiological responses, very different projections to different parts of the brain, but there's no way of telling them apart unless you do anatomy or unless you do some biochemistry. And so the idea is to do that on a large industrial scale in the same way that the genomics project was able to scale up the sequencing of the genome. We just can't afford to do this piecemeal. We want to be able to do that completely and extensively in every area of the brain. We've already heard about advances in optical recording. For example, we're now able to record several thousand neurons at the same time. And our goal is to increase that over the next 10 years to 1 million neurons at the same time. Now this becomes a real problem, and you'll see in a minute. it changes the nature of the questions you're going to ask. Connectomics. You need a wiring diagram. Now, when this issue was being debated just a few years ago, there was a lot of skepticism. Right? There's a lot of skepticism about, if you just have the wiring diagram, does that really tell you how the brain works? And of course it doesn't by itself, but it turns out if you do want to know how the brain works and you don't have the wiring diagram, it's like trying to swim with your hands behind your back. You've got to, once you have that in conjunction with physiological recordings and with biochemistry, it's the integration of all of those data sets that will really be what's necessary. And this was brought up earlier, which is to say, there's a revolution also in behavior, that we need to be able to design experiments that are going to take advantage of these techniques. And the simple behaviors that we study now, in most of us, like, choice that you give an animal to do, forced choice. You get a monkey, a very complex cognitive creature, you put him into a head-fixed chair, and you train him for 6 months to make a decision between two objects, right? Is that the way that the brain it evolved to do that? No, I mean we simplified it for practical reasons, but the fact is we have tools and techniques now for being able to get the monkey out of the chair, basically. So that requires a completely different way of designing experiments. And then finally, and this is the part that I was very pleased was included, in fact, amongst the major recommendations, was that I think for the first time in the neuroscience community, there's a general agreement now. Not everybody believes this, by the way. There's a lot of diehards out there who basically don't think they need any theory or modeling. But the fact is that we need more and more as we collect more and more data, and because the questions get harder and the analysis gets harder. And so this reminds me of a similar transition that occurred in molecular genetics. So about 15 years ago, I was asked to give a talk at a molecular genetics meeting at Cold Spring Harbor Lab. So this is really hardcore molecular genetics. I was the only person there who wasn't doing molecular genetics. And so I was, why do they want me to give a talk, and what could I possibly say to them? And I was very fortunate because the speaker just before me was Lee Hood. So Lee is a legendary molecular geneticist who just at that time was moving from Caltech to Seattle, where he set up a whole new systems lab and institute. And here's what he said. He said, I got up one morning and I asked myself, why is it that I have more computer scientists on my payroll than I have biologists? And this is the early days of bioinformatics, right? And he realizes that, well, computer scientists know a lot more about information than biologists. Now, that got me thinking, okay? Because he put his finger on something, which has to do with the state that different fields are in. So if you look at biology up to about 20 years ago, it was all about reaction kinetics. It was all about biochemistry, about energy, about reactions that took place inside cells, cell biology, and trying to understand in disease, when those reactions go wrong, when there's mutation, and so forth. And the shift that occurred there, of course, was the structure of DNA in 1953. And the thing about DNA is that not only is it a chemical, not only is it have energy reactions and so forth, all that is essential to the function of DNA, but it also carries information, symbolic information, that is used by the cell in order to direct traffic and create new cells. And information has a different status than the energy, and it really involves different ways of thinking. Now, what organ of the body has more, whose function has more to do about information than the brain? Right? That's why it evolved. It evolved to take in information, sensory information. It evolved to hold on to the information. It evolved to be able to make decisions, to be able to move And the motor system is absolutely incredibly important. You're going through a bottleneck. You can only make one motion at a time, so you better choose wisely. All that has to happen in real time. So it's really the organ that for which we really need to get help from people who have been trained in these areas: theory, modeling, computation, and statistics. So that's really what I'm going to tell you about today, is a few things that are going to be happening with regard to this particular recommendation. Now, what's common to all of these different recommendations is that it's all about scaling up. It's all about taking what's been done one neuron at a time and scaling it up to many neurons. And I'll give an example of that in a second. This has already appeared twice in this meeting. This appeared just about in November, actually, so last month. It's amazing how fast these things spread. I just want to make one more point here that wasn't made, which is that, see this little inset here? This was the version that Pat Churchland and I published in 1988. You'll notice that it's a lot sparser than this one, right? So this is what 30 years of research developing new techniques will do for you. For example, fMRI did not exist. See this big hole here? Well, what's happened is that over the years, these holes have been filled up. And I'm sure it's going to continue to fill up, but that's really what's going on, is that as new techniques get developed, and we're trying to accelerate that, we'll have more and more ways of being able to understand what's happening at a particular spatial and temporal scale. Okay, so let's just go through TMCS. This is an unfortunate acronym because you can't really pronounce it. TIMCS, perhaps. And let's start down here at the bottom, okay? So, and I should say that the three of the people on the committee that were responsible for this, tremendously helpful colleagues of mine, was Emory Brown, who is a background in statistics. He's actually an anesthesiologist at Harvard. And Eve Marder, who is a invertebrate neurobiologist, but is a born-again modeler, in the sense that she has cultivated relationships, collaborations with Larry Abbott and others, and really has it integrated into her own research program. And I think she's a model for how this is going to go forward in the future in terms of teams and collaborations. But what Emory really emphasizes is that especially as these experiments get bigger and more complex, it's too late to come to a statistician after you've collected the data and say, could you analyze it for me? Because you may not have made the right measurements over the right time period in order to be able to answer the question that you wanted. You've got to have this design. The statistician has to be there at the very beginning when you're actually setting up your experiment. Otherwise, you may have to do it all over again. So that's a very important. We were just talking about statistics at the table just before that. Right now, if you go to take a statistics class in psychology, you learn how to do a t-test, and this is really not the right way to begin training our students. We need to be able to train them on sophisticated ways of using the best approaches that are available. Computation means a lot of different things, but at least in the report, we focus on things like how is information in this, for example, the visual scene, how is it encoded into spike trains? How are those spike trains transformed in the retina and then into the cortex? And how is that information decoded? So these are all questions that people in information theory struggle with all the time. And so there's a lot of tools there. Modeling. Well, models, there are many different kinds of models. And that's the key thing, is no single model. You need a model at each of these levels, and at each of the levels you have models of different granularity. So you have to be able to take the data that's available and basically model, and this is something that we're doing in my own lab, is what you're really doing is modeling the experiment. You're modeling the actual process, because you often have to take into account the limitations of the actual measuring technique. In one example, you saw beautiful pictures of calcium signaling, with this is GCAMP6. Well, it turns out that GCAMP6 is a calcium buffer. What does that mean? That means that when the calcium comes in, it binds and it fluoresces. Well, that means that the free calcium is not going to be the same as it would be if the indicator wasn't there, which means that, it's, in physics, it's inevitable that, your instrument that you're using to make the measurement is actually modifying the actual process itself. That has to be taken into account. And so what you do is you model with the indicator, you match the data, and you take away the indicator from the model, and you see what the result is if it hadn't been there. And that's the ground truth. It's not what was being measured initially. Now, this theory is the most difficult part, in part because we don't have any good theories about the brain. We have lots of theories, but I don't think we have any really good ones. And that's because I think that theory in biology has a different character from theory in other areas of science. So what has been the most successful theory in biology?
Evolution.
Evolution. Okay. Well, let's think about evolution, okay? Is it predictive?
No.
It's not really in the same sense that a physics theory is predictive. It's not like you can set up an experiment and test it, right? I mean, a lot of it it's a beautiful theory, and I have to admit that it's conceptually been very essential. And as Sejnowski once said, nothing in biology makes any sense except in the light of evolution. But the fact is that evolution itself is something that is really a different beast from what we would call a theory in physics. Okay, here's another. Okay, what is the best model in all of biology in terms of modeling? Well, if you're a neuroscientist, that would be correct. But there's more to biology than neuroscience. Okay, so when Watson and Crick looked at the structure of DNA, they actually built it out of metal plates. And there's a famous picture of them pointing up to the way that the base pairs fit together. And I have to remind you, that was a model, right? But what's interesting was not only was it a model, but it was also a theory for how information can be passed on from one generation to the next. And it also was a model for evolution, mutation. So this is the kind of theory that has had an impact in biology, and we're still searching for that kind of a model in the brain. And I was privileged to be a colleague of Francis Crick for many years at the Salk Institute, and we would discuss this issue. What kind of a theory would really be essential to making progress? Francis's approach, and it's all personal, right? His approach was to look for inspiration, theoretical inspiration in neuroanatomy. He knew more about the anatomy of the visual system than any other person I know, because he read all the papers and he talked to all the anatomists, and so he knew the very fine details. And I often wondered, why was he taking that as his focus? And I realized, well, it was successful with DNA. Maybe it'll be successful by looking at the structure of the brain. That the structure of the brain may be telling us something. And he didn't live long enough to see connectomics, but I think that there may still be a lot that will come from that approach. And I remember one day at tea, he would come to my lab every day for tea, English tradition. And he said, "Terry, you've gotten too fond of your models." And I thought for a moment, and I said, "Well, but they're really good models." He said, no. The model is not an end in itself. And here's what you should be thinking, how you should think about it. Purpose of a model is to design a killer experiment that no one else would have thought of doing without the model, which, if it's successful, will give the game away. That's what we should be searching for. Let's just not go out willy-nilly and just collect data at random. That's what we've been doing for forever in neuroscience. But it has to be directed. We have to have the right questions that are going to help focus on which data are the critical ones that we need in order to give the game away. And that's really what the goal is going to be. Now, this came up earlier, and I was going to emphasize how important this is, which is that a whole new generation of neuroscientists now need to be trained with skills in statistical reasoning. They should be able to write a simple MATLAB program. If they can't, they're not going to be successful in neuroscience because they're not going to be able to analyze their data. You know, it's becoming obvious now. Actually, a lot of students are already doing that, but our curriculum is way behind, and the way that we think about the education for students in neuroscience, we need summer schools, we need a better mentoring. By the way, this all happened in molecular genetics a generation ago. They have a whole area now called bioinformatics in their courses, and my colleague at the Salk Institute can't do any of this, the bioinformatics, but they have students who can do it for them, right? Because, you know, they were successful at a time when you didn't need to be able to run, you know, all these search engines and browsers and so forth. And this is something that Eve Martin really is emphasizing, which is that you're just not a user. You should be building. You should be there building a model of your own data. Okay, now I want to step back and just give a few examples of what's happening, and you've already heard some. So this is a famous picture from David Hubel. This is a tungsten microelectrode. This is a Golgi stained piece of tissue. So this is really, it's Photoshop before Photoshop. This paper in '62 was incredibly influential in guiding 50 years of research. Because what Hubel and Wiesel showed is if you record from single units in the visual cortex of the cat, initially, and then the monkey. By the way, why are they called units, not neurons? Because they couldn't prove they were single neurons. But they heard, because if you see a spike, how do you know it's from the same neuron? But in any case, they called them units. And they showed that the response properties are very precise, that they're oriented, and they have specific properties in terms of responding to the onset and offset. Now this paper launched 1,000 microelectrodes into the brain. It was such a beautiful tool and technique. And you know, you could do this anywhere. Auditory system, somatosensory system. Just march into the rest of the brain, and that was a 50-year program. And it's still going on. Vast majority of the labs are still doing this. Okay, so I want to make an analogy here. Suppose that you knew nothing about vision. In fact, you know, the only thing you could see is a single pixel, like looking at the world through a soda straw. So you could look around one pixel at a time. You know, you have a megapixel image, and you've got to figure out what's in that image, one pixel at a time, right? It's hopeless. You'll never understand the global organization by doing it one pixel at a time, and that's what we've been trying to do. You need to be able to open it up and look at enough pixels so that you get a sense for, you know, what is the structure in the image and what is it that constitutes an object. And this is something people in computer vision are struggling with. But the fact is that we're still at this stage, and it's not just the low data rate. It's the fact that you start thinking and theorizing in terms of what you see at the tip of your electrode. You know, you've heard about the grandmother cell theory. So the most popular theory, still alive, is that as you go up the visual system, the cells get more and more specific, and Jack will, I hope, corroborate this. And at some point, you will find a neuron in your visual system which will only respond to your grandmother. And I think this is we sometimes, someday in the vast future, it'll be called the microelectrode theory, because it's based on the idea that all you need to know is what's at the end of the tip of a single microelectrode. Okay, you've already seen this. This is a beautiful work from Misha Aaron's lab, and now being this is a snapshot from a paper that appeared in Nature Methods just recently. The point I want to make here, you saw the beautiful experiment where they varied the direction of motion, and it's color-coded here. What I want to point out is that this is a very, very simple stimulus, just a moving bar moving in different directions. Almost the entire brain is activated to one extent or another in a very lawful way. And this has come out over and over again. So the way that a neuroscientist, for example, without this technique would go about and say, well, we know that the tectum is the place where the information is converging, so we're going to record from cells here. Well, you're ignoring 99.9% of the rest of the cells, and how do you know whether the cells you've picked out are the important ones? In fact, maybe they're all important. So that's what I mean. We need a new theoretical framework for thinking about global interactions. And this, by the way, often techniques can bias you and distort your views. And an example of that is brain imaging. Again, a very powerful technique. It's non-invasive. We've learned a lot. But unfortunately, if you look at these beautiful pictures, with these hot spots, you will get the impression that, whoa, that's the area where faces are recognized. That's the fusiform face area, because that's where you get the... Well, what's happening is that there's activity over the whole brain when you give a face, but if you threshold it, you'll see just the tops of the mountains. And why should we just be looking at the tops of the mountains when we know that there's activity everywhere else? So that's where we're coming from. We have to be able to look everywhere else and understand how all that information gets integrated together. So I have to show you this one too. And I'm going to play it. And I want you, so you saw the beautiful experiments, okay? I want you to look at this pattern and tell me what was the larva zebrafish doing? What was the behavior that was giving rise to this pattern? You see a lot of the same neurons are being activated that we saw before that were directionally selective.
Meditating. That's sort of true.
So you're absolutely right. It turns out that this is an immobilized zebrafish that is doing absolutely nothing as far as we could tell from the outside. But it's clear that it's doing something on the inside.
He might be trying, he's probably trying to swim. He's immobilized, but imagine being trapped and trying to move. That could be something what's going on here.
Okay, so this illustrates a problem that we have. Up until recently, the only thing we could possibly do was an experiment where you control the stimulus and you look at the behavior, right? That's turning a very complex cognitive creature into a reflex machine, SR machine, right? That's not, of course, you could do that. You can train monkeys, but that's not what the brain evolved to do, right? Intrinsic activity can spontaneously be generated. In fact, when you sit back and close your eyes and think, that's spontaneous activity. And the problem is there's no way to correlate it with anything outside to give you any sense for what it might be doing. But that's where we need to go. And I think I'm going to skip over this very quickly. This is a really nice experiment from Bill Newsom's lab. And I'll just tell you the bottom line. If you do an experiment that involves only a few different sensory choices, in Bill's lab it's motion to the right or left, it doesn't matter how many neurons you record from. You can record from 10 neurons or 100 or 1,000. You can PCA down the activity down to 2 or 3 dimensions and capture 90% of the variance, right? What is that telling us? Is that telling us that all these neurons are being wasted? No, it means that your task is too simple. The dimensionality of the task is what's limiting the dimensionality of the brain signals. And the solution for that is to increase the dimensionality of the task commensurately with the dimensionality of the data, the dimension of the data that you're able to record from. So if you're recording 1,000 neurons, you should have a task that has 100 dimensions or 1,000 dimensions. So another thing that's happening is that this is something that Sidney Brenner was very instrumental in bringing into biology, the idea of completeness. So he, very early on with C. elegans, realized that if they had a complete list of all the cells in a simple creature and also a complete wiring diagram of the brain of the creature, that would really help us understand a lot of things like development and behavior. Well, behavior has taken a little longer because you couldn't record from the cells. But it turns out it's been a gold mine for people who are studying how cells develop and divide and how they become wired together and so forth. And what we could do now with the tools and techniques that are becoming available in anatomy is doing the same thing, completeness. And this is an example of work that's being done by Krishnan Padmanabhan, who's a Crick Jacobs Junior Fellow working with my lab and also in collaboration with Rusty Gage and Ed Calloway at the Salk Institute. So this is the olfactory system in the mouse. This is the olfactory epithelium. A mouse has about 1,000 different receptors that project to the olfactory bulb. There's tens of millions of fibers projecting into the bulb, and then from there the mitral cells project into the piriform cortex and the accessory olfactory nucleus, and several other places, but these are the main ones. The actual pattern, the divergence and convergence, is really interesting. So it turns out that it's salt and pepper. Each one of the colors is a different receptor for recognizing different classes of chemicals. But all of the red ones converge on the red glomerulus, and all of the yellow ones on the yellow glomerulus. So it's a tremendous convergence going on. But what happens, though, as the mitral cells project into these other structures, you can see that in the piriform cortex, it becomes salt and pepper again. That means that if you look at any little region here, you'll be likely to be getting inputs from any number of these, typically on the order of 20 or 30 glomeruli for a single pyramidal cell. But if you look in the accessory olfactory nucleus, it's a topographic map. So different parts of the brain are decoding this information in different ways. What's interesting here is that if you think about the subspace, each one of these little patches here is sampling a subspace of olfactory space. You know, if you have 1,000 receptors, 1,000 dimensions. You know, color is done with 3 cones, so that gives us 1,000,000 different distinguishable colors. You can imagine with 1,000 receptors how many different odors you can recognize. But the point, though, is that you have no idea which combination is going to be important for a particular behavior that you want to avoid, you may want to be attracted to it. And the idea is each one of these little patches here is sampling a subspace of odor space, very high dimensional space. But something else is going on here. Now, that's not the whole story. This is the feed-forward story. Every one of these projections going forward has a feedback projection coming back. Now, what's going on there? Well, we don't know. These are the most difficult projections to study or analyze. And I'm not going to go into this. People in the visual system have struggled with this for a long time. And it's even more mysterious in the olfactory system. And so Krishnan used a technique developed by Ed Calloway to answer the question, I want to map every single neuron that projects back to the bulb. All the feedback connections to the bulb, right? The bulb is like the retina in many ways. It's kind of where the first representation of the olfactory world exists. So the technique that was used was to inject a virus, as shown here in green, into the granule cell layer of the bulb. And that's taken up by the local neurons. But the virus, it's a rabies virus, which actually is able to jump transsynaptically to axons that are projecting into the bulb. And those then project back to some other area. And these viruses were actually engineered so that they don't jump again. It's by changing the coat so that they can't get past that. After the first one, they can't get past the second one. So now you wait a week, and now you can slice the brain up, and you can reconstruct every single neuron that projected to that point where you injected the dye. And this is what it looks like. So obviously, you're around the injection site, you're labeling a lot of neurons, but you label. You can see from a local injection, you're getting a huge area of the piriform cortex, accessory optic nucleus, the diagonal band. It's a very important neuromodulatory area with acetylcholine. And then, and these are all known, but now we can actually count them and get a more quantitative measure of relative numbers of neurons that are projecting back. And then there are a bunch that are shown here in red that have never been seen before. What are they? Where are they coming from? Let me first show you what the reconstruction looks like, and it's hopefully will give you a. Oh, okay. Let's go back. Okay, well, I had a movie that basically takes this into 3D and turns it around, but I can tell you that a lot of the neurons that are, here's the front of the brain, this is the back of the brain, turn out to be emanating from the hippocampus. So the hippocampus is about as far as you can get away from the sensory periphery. All the sensory systems funnel into the hippocampus. And the hippocampus has feedback projections. And it's known that if you damage this part of the brain, you have trouble with memory, long-term declarative memory of events and objects. And this has been extensively studied in humans, and we have these famous cases like HM, and also been studied in rats. But here's this, a pyramidal cell, C1 pyramidal cell. Now, who would have guessed that this pyramidal cell, at the very top level of analysis, is projecting all the way back to the first level, back into the olfactory bulb? What's going on? Well, we can get some clues by looking at behavior. So there's a famous passage in Proust's novel, À la recherche du temps perdu, in which he describes, as an adult, tasting a Madeleine and being transported back to his childhood, where he associated this taste and smell with an episode, and he describes how evocative it was. It was like he was back in a time machine, going back to his early childhood. And olfactory memories are like that. They really are very, very powerful. So here we have something very, very interesting, is that there's this two-way communication going between the lowest level and the highest level. And this may have something to do with the fact that we're looking at a global system. We're not looking at a system that's doing local analysis all the way up. At some point, the global and the local have to mesh, and that's one of the big challenges. How much time do I have left? I should be wrapping up.
Yeah, a few more minutes for questions.
Okay. Okay, so I'm not going to say much about DARPA. But I will say a few words about NIPS. Okay. So I've emphasized one of the themes of this talk has been scaling up. It turns out that a lot of algorithms don't scale up very well. A lot of the AI algorithms that worked really well in toy worlds, when you scale them up to the real world, they just crash and burn. Not because they were bad algorithms, but because they just were exponential. They were NP complete, right? Well, it turns out that if you have really, really big data sets, even polynomial algorithms crash and burn. So you really are looking for low-dimensional algorithms, that scale with n or square root of n or log n. That's what we're looking for. And very likely that this is the kind of algorithms that the brain has, nature has discovered in the brain, because the time complexity is very short. You've got to make a decision within 100 milliseconds. Tremendous amount of data is coming in. You've got to make a decision because your life depends on it. So this is a challenge. How do you characterize the complexity of the way that the brain is solving the problem? And the internet companies have the same problem. They have huge databases they've got to search, and they can't afford an algorithm that's going to take a minute. If it's more than 1/10 of a second, forget it. It's got to be, it really has to be almost like a reflex. Now, of course, after you've reacted, you have time to think. Maybe the feedback connections are giving you the second chance to go back and reconsider what you've just done. And so now you get into the recurrence, and that's another part of the system, is that it responds, but it also is able to reflect. Now, starting on Monday, the NIPS meeting stands for Neural Information Processing Systems. This was a group of neuroscientists, primarily, and I think Todd, were you there at one of the first meetings? Todd Lane?
Yeah, going back to, I have the red book. What year was that?
Yeah, that's the NIP book. That was 0. We're up to like 28 now. Jan was there. Yep.
I missed one.
Okay. Okay. So it turns out that a lot of the algorithms that were developed by that community now have had a really major impact, are having a major impact on all the internet companies, and interestingly, on almost every area of science with big data, because of the fact that we started out with our problem being handling the brain, which is the ultimate big problem, with understanding difficult problems in speech recognition, image, object recognition, translation, and just within the last 2 or 3 years now, it's been possible to scale up those, roughly the same algorithm. I don't think they've changed that much. I know that Jan's convolutional network has more bells and whistles, but what's really changed over the last 30 years has been that the power of computers has increased by a factor of at least 1 million. The amount of data that you could use has gone up even larger, all the images on the internet. And those two together have really have taught us something really important, which we didn't know, which is that these learning algorithms scale. They scale with the size of the network and the amount of data that you have. We didn't know that back in 1980, in the 80s. We had very small networks with just a few, 1 layer of hidden units, and now we have 12 layers of hidden units. And just this is one of these Google. I'm not even sure it was an experiment, but in any case, they put 10 million YouTube images through one of these deep networks, and they found units at the early stages that look a lot like simple cells in the visual cortex, and then face cells in the middle of the cortex. A lot more cat cells than face cells, but that's YouTube.
I should point out that this model was completely, because complete deadline was abandoned, a few months after it was published. It doesn't work.
Okay, well, talk to Jan if you want to know what works.
Convolutional net.
Okay, so let me just end. Was that a convolutional net they were using?
It was not convolutional in the sense that weights were not shared across locations, and it was trained and supervised with a technique that basically doesn't make any sense.
Oh, okay.
So it doesn't really work.
But it still found cats.
Well, but any stupid thing will find cats. So it's like it's like one of the principal components, right?
Okay. It's an interesting. We should try PCA on YouTube. Yeah.
It's an algorithm.
Right. Okay. So I want to end with the following thought. One of the criticisms that we had and still have is that in the BRAIN initiative we couldn't define a target like John F. Kennedy said we're going to put a man on the moon. Well, what's the equivalent of the moon for the BRAIN initiative? How do we know when we get there, right? And it's a difficult question because, and we debated this endlessly, and I don't think there's any consensus on this. But what I did finally realize, after a lot of disagreement, I thought maybe you should have enhancing memory as the goal, and somebody else said no, paraplegics need to progress too. But what I realized was we're asking the wrong question. So if the moon was so important, why haven't we gone back since 1969, right? And that's because it's the MacGuffin. Hitchcock film, he had something which everybody thought was really important. That wasn't what was important about the moonshot. It wasn't the moon itself. It was this symbology of going to the moon politically, but it is also a much more important economic investment that was made. $100 billion was invested in the American economy in training engineers, in developing better materials in order to be able to come back through the atmosphere, integrated circuits that would allow us to compute information in real time as the astronauts were circling the moon. Rockets, nobody could build a rocket big enough to go to the moon in 1961 when the announcement was made. And here's an interesting factoid. The average age of an engineer in 1969, when Neil Armstrong actually stepped foot on the moon, was 26. And what that means is that 8 years earlier, they were all in school. This, I think, is by far the most important thing that has come out of all of these major projects, going all the way from war on cancer the human genomic project, and the BRAIN initiative is that this is an investment in new technology that is going to have an influence on the economy. For example, the Human Genomics Project. The Battelle Institute did a study. For every dollar that was invested, which is about $5 billion, 4.5, and I think it was about 3 to 3.5 billion. For every dollar that was invested, there's $140 in economic activity that was generated, not just in the healthcare industry, but also in agribusiness, in ways that nobody could have predicted. And in the case of the war on cancer, we haven't beat cancer, but it created a biotech industry based on recombinant DNA technology. So it's really the investment, not just in the technology, but it's an investment in intellectual capital, into the engineers that are going to be trained, and the neuroscientists going to be trained. In other words, a whole generation are really inspired now. They really think that they can make a contribution. And what's really, for me, very, very satisfying is that a lot of I talk to some of these young people when they're making their decision. They come to me, they ask me, what's going to happen over the next 10 years? And a lot of them who would have gone, these are really smart kids graduating from Harvard, who would have gone to investment banking are actually going into the BRAIN initiative. They're going into neuroscience. And they're coming from engineering schools. They're coming from biological backgrounds, from mathematics. So I really think that this is an opportunity for the nation to do something really good for our, for the future, for the problems that are going to be paramount within our lifetime. As Alzheimer's climbs, as we age, Alzheimer's becomes more prevalent. All the mental disorders, you saw the figures. It costs much more to handle all of the mental and neurological disorders. And the reason is that these are people who don't die promptly like they do from heart attacks or from cancer. These are people who live often many full lives, like schizophrenia patients, and have to be cared for. Alzheimer's, it's just devastating. It's not just the healthcare system, it's the families, the burden on the loved ones. And so if we don't do something now, make an investment into the future, we're just going to be saddled with even more burden up the road. So that's the end of my talk. Thank you.
I have to confess I'm floored. I have proposed that we continue the discussion in the break.
Okay. Actually, I would like to make a quick comment and a question, if I may. I know that, if I may, I know that we're late. Very quick comment.
We're going to cancel the break.
So,
we already had the break.
Sorry,
Yeah, we moved the break.
Okay, all right, cool. Yeah, cool.
So,
We'll take questions.
Terry, I have a quick comment and a question. My quick comment is about NIPS. As one of the founding mothers, I guess, of NIPS, I would like to say that I very much regret that NIPS has actually become IPS over the years. It has lost the N, and it's no longer true that it's a conference of neuroscientists. It has become the premier modern statistics conference. And I would like to challenge you and other people who are very active in NIPS to bring the N back, because I haven't been to NIPS in perhaps 10 years now, because it doesn't reflect my interests as a theoretical neuroscientist. So in the spirit of this meeting, of bringing computer science and neuroscience together, I would like to encourage you to do something about it. Now my question, my technical question.
Why don't you come this year?
Say it again?
Why don't you come this year?
We have other conferences, there is very little neuroscience in NIPS. I stopped sending my students because there is no audience for their papers. So you made a very interesting comment about task complexity and about how if one wants to record from 1,000 neurons and see the need for 1,000 neurons, what should design tasks whose complexity or dimensionality involves that many degrees of freedom. And I would like to ask for comments on what do you think about primary motor cortex in that context. Primary motor cortex is controlling a plant that has at most 100 degrees of freedom, a few hundred degrees of freedom, which is the number of muscles in the body. So how do you envision, in the context of actuating muscles, a task that would involve the complexity that required millions of neurons?
Cross products, yeah. No, let me start from your comment about NIPS, because I think it's a very important one. So first of all, I just got an email from somebody who hasn't been to NIPS for a long time like you, saying, you know, the N is back in NIPS. And that's because these neural network models that Jan has been working on for 30 years now have outperformed all the other machine learning classifiers in these practical problems. Now, the issue that you said with regard to neuroscientists, of course, this is not a neuroscience conference. And people wouldn't come there if they wanted to learn about neuroscience. But what is happening is that the people in neuroscience who need the tools that are being developed, they are the ones. Liam Paninsky is coming, people who are statisticians. And it's true not just for neuroscience. Every single group is in the minority there. The computer vision people, only 5%. The people from speech, 5%. The mathematicians, 5%. The physicists, 5%. This is not the main meeting for any one of those groups. You don't go to NIPS to learn physics. You don't go to NIPS to learn how to do computer vision. But it's had a huge impact on computer vision. In fact, it's reached the point now where CVPR is actually complaining that, that papers to our workshops are competing with their papers or being presented there. So the important thing is that all these tribes that have their own sectarian problems and issues, not everybody in those areas, not everybody in computer vision, not everybody in neuroscience, need to come and develop the tools. By the way, it's also areas like astronomy that we heard about. They have computational problems too, and it turns out that the very same algorithms that you can use for speech can be used for astronomy. So that's my little comment, and I agree with you that we need to get more. The N should be bigger. Maybe it should be capitalized. But I'm just saying it's really historical that the N is there, but it is coming back. Now, with regard to, what was the other question?
How do you design motor tasks that with so many degrees of freedom?
Oh, okay. No, so this is interesting. So this has to also, this is a beautiful question having to do with complexity. So almost all the work that's done in psychophysics, human psychophysics, Georgopoulos and in monkey recording, and also in robotics, is done with just a few degrees of freedom. Typically, you have the animal moving a manipulandum in 8 different directions, right? And then you record from cells in the motor cortex and find, lo and behold, there are cells that are tuned for different directions. So this has really been a problem. In fact, as people do different tasks, they find that there are cells that are selective for the dynamics of the torque and so forth. And actually, it's gotten to the point now where if you go to a motor meeting and ask the question of what's being represented in the motor cortex, people will say everything, and that's not a good answer. However, one thing for sure we know is that a lot of them have this cosine tuning curve. That's something that Georgopoulos showed. That is to say, as you move your manipulandum, just like you saw with the zebrafish, there's one particular neuron will prefer a direction of motion. Well, it turns out that there's a motor neuron that prefers that direction of motion too. The trouble is that I think that result has been misinterpreted because it's really telling us something about what geometry is the brain representing. There's 2 different choices. One is intrinsic coordinates like joint angles, and the other one is a coordinate system that is fixed in the world, a spatial coordinate system that's extrinsic. And if you just write down the equations of motion, it turns out that they're much simpler if you have a fixed reference frame than if you have a moving reference frame. And you get, the number of terms grows exponentially as you add the number of degrees of freedom. And that's why people have only used 1 or 2 or 3 at most degrees of freedom. But the fact is that every time you reach for something, every single joint and muscle in your body is moving to compensate for your balance, for all of the fine details that have to be done to grab it. And that means the brain is working in a very high dimensional space. And we're not designing experiments that tap that. We clamp the monkey down. I told you this, and we just let it move a few of the dimensions. We need to be able to open up the gate to be able to look at the behavior, find out, and quantify all the dimensions of the body, not just 1 or 2, and then see how those are represented, all those dimensions, in the motor cortex. So that's I guess, one way to go. But you know, I don't have the answers. I think the motor people really, it's an opportunity now for the motor people. And actually, as you know, it's now possible to do that not just in monkeys, but in humans. So there are people walking around now, these are quadriplegics, not walking, paraplegics, who can't move their arms, but they, through an implant into the motor cortex, recording from 100 neurons, this is John Donahue and Andy Schwartz and others, they can translate those signals into a robot arm that can pick things up and move them around just by thinking. And actually, you know, there was a famous video of someone picking up a cup of coffee, and for the first time since they had their accident, they can actually drink coffee. So this is it's a whole new era now, and I think we're just beginning.
Can I quickly?
Couple of quick questions.
Yes, but this is. I think this is a very, very nice point, but there is a fact that I think is related to, for instance, in the motor system. One aspect is space, but another aspect is time. And for instance, the number of neurons that you have, for instance, in spinal cord, is largely also related to the way in which you know, these neurons happens into an orderly sequence in time. So, and going to that, I think one of the issue in your nice graph about space and time in neuroscience, those are, even in terms of the location of each one of us in this graph, it's interesting because there are two different dimensions. I think maybe I'm wrong, but my feeling is that while we are somewhat specialized in the space domain, you can work at the level of synapses or the level of behavior. I think that we have, in general, a relatively large range of time scales. And it seems to me... Huh? Quick, it's finished. The question is that I would like to know how you would deal with this relative scale of or relative importance of time and space in organizing you know, interactions between neuroscience.
Well, I think that you're right. I mean, it's really interesting. I sometimes put this diagram up when I'm talking to a broad audience, like, you know, first-year recruit graduate students, and I ask them, you know, take a pin and put yourself up there. You know, where do you want to work, or where are you working? And, you know, they cover the map. But occasionally you find a student who said, well, do I have to put it in one place? Because I really would like to do this, I'd like to do that. They're actively discouraged from doing that. We have set up a system where we incentivize specialization. And so our students are, you know, discovering more and more about less and less. And that, if you go to a neuroscience meeting, it's really overwhelming. You have a sea of thousands and thousands of posters, each one of them, you know, a little micro contribution to a particular area of the brain, a particular neuron, a particular, you know, neurotransmitter. And sitting next to their poster is somebody who's doing something that is a, you know, what's the connection? I mean, so I actually think that we beat it out of. I think our students coming in actually want to be able to integrate across levels, and they're told that no, you know, if you want to do a thesis, I'm sorry, you're going to have to work on that molecule. So here's a little anecdote. Okay, this comes from Jeff Hawkins. So, you know, he's the guy who started Pilot and you know, first PDA. Well, he started out as a graduate student at Berkeley, and he went to Frank Werbeling's lab, who works on the retina, and he said, I want to work on associative neural networks. This is back in this, you know, early 80s. And he said, I think that we can make progress understanding how, you know, neurons communicate with each other. And Frank said, well, we don't work on associative networks in this lab. You know, if you want to get a PhD, you've got to work on my neuron in the retina. And here's the experiment you're going to do, because that's what I have a grant to do. And if you want to work on associative network models, then, you know, do that after you've graduated, because, you know, I don't have any money for it. And, you know, and really, a lot of students come in and want to tackle big questions.
That's why all of us walk to the Palm Pilot on our projects, right? Or on our pockets.
Yeah. Yeah. So what happened to him? Well, he had 2 kids. His wife said, "You're not making enough money as a graduate student." So he went off into industry. You you know, everyone told him that, you know, the PDA is never going to work. It's been bombed at Apple, and if Apple can't make it work, nobody's going to make it work. And he made it work. He you know, used something called graffiti to be able to communicate with it, and it was a very primitive device, but actually it sort of had the right form factor, and it really started the whole revolution. Well, what is he doing now? He has a company where he wants developing models of associative networks. He's come full circle, but you know, that's the rare case.
We have one more question.
I wanted first to comment and then to ask a question. So, wow, I feel like in a computer science meeting, not in a brain meeting now. Okay, so it's actually very heartwarming that that person said in front of you that he has lots of computer scientists on his payroll. And I was curious how many computer scientists were in this NIH 2025 meeting when you decided what to do with the brain.
Well, on our committee?
Yeah. How many PhD in computer science were there?
PhDs in computer science. I don't think there are any.
How many PhD in computer science do you have on the payroll in the Salk Institute?
You know, quite a few. You know, so if you know, loosely speaking, in terms of systems administrators, in terms of PhDs, you know, probably two or three.
Okay.
But I think you're making an important point, which is, and this is exactly why the Brain Initiative is I think going to change. It's a cultural problem that we're dealing with. It's not just a technical problem. It's just not, you know, getting a better gadget. It's how do you get the better gadget? Well, you have to get the computer scientists working together, cheek to jowl, with the neuroscientists. They actually have to collaborate together, and it's very difficult, as you know, to get you know, if you're in a biology department, to hire a computer scientist. It's beginning to happen. It's like in bioinformatics, it's already happened. A lot more, I have to say, okay, in my little area, computational neuroscience, a lot of people have backgrounds in physics and math and computer science. They're getting jobs now. In fact, it's maybe partly through the Brain Initiative, but the fact is that this is a growing market. That departments that traditionally wouldn't hire my students now are asking me, you know, do you have any good students coming out? Because they really think that this is going to be important for the future. Yeah,
and I just kind of wanted to give you a compliment here in front of other people. So in the years that I came to visit Francis Crick in the summer at the Salk Institute, he really saw your lab as one of the most open one in terms of understanding that computer science is going to affect neuroscience, where you were, you know, this was many years ago. And I think this is one of the things that is still in the field, that there's still neuroscientists that think of computer science as writing the program, and the idea of information or algorithms to give to physicists who they consider expert in computer science.
Actually, now that you mention it, our last hire was a computer scientist from Carnegie Mellon University, who's working on complexity and actually thinking about the following problem using graph theory. So the question is, you know, the brain develops. It starts from cells that have to connect with each other. And the question is, if you want to end up with a certain connectivity, is it better to do each of the connections precisely at the beginning and just build it up? Or should you be more exuberant and make many, many more connections than you need and then prune away? So he actually solved that as a computer science problem, and then he went in and tested it. But he actually went into the lab and actually did some experiments, anatomy experiments, and showed that that's actually what the brain is doing. So, so there you go. So this is, it's already happening.